and Professor Rowitch has kindly agreed to talk today on early genetic testing in children with developmental and medical complexity. And it's going to last around 45 minutes with some Q&A afterwards. So many thanks, David, and over to you.
Okay, thanks very much, Ben. Thanks everyone for joining, and I hope we'll have time for some discussion afterwards. I was asked to talk about sort of the way that we can use genomic tools in pediatrics, thinking about kids with complexity, and I'll focus on some presentations of some neurological presentations today. So some general considerations about genome medicine and why this makes sense to come into pediatrics. There are statistics from North America that suggest that genetic diseases could account for 30 to 40% of admissions to hospitals, and also can be the cause of a lot of the costs, because these are kids that will have complex needs and have repeat hospitalizations in many procedures. We know that rare diseases can account for a large percentage of death in the first year of age, and that unfortunately 30% of children with a rare disease can die by their 5th birthday. So how can we use genomic tools to improve diagnosis and inform clinical care? That will be one of the challenges, I think, that as a hospital-based pediatrician, I'm a neonatologist, will be very interested to see. So we think that genome medicine can help with our health provision through diagnostics, precision therapeutics, and then maybe better prediction so we can counsel families and really anticipate future complications. And with a lot of discussion around precision medicine, it focuses on adults, which I think is illogical. We should be thinking about applying these tools early when they can really have life course impact. And that really helps to foster a prevention perspective that I think more and more will come into the practice of pediatrics. But there are sort of limitations, and we have to be cognizant of the sort of current state of affairs. Generally, genomic medicine is not practiced in pediatrics, and this limits effective detection. There are challenges such as we only understand a fraction of the information in the genome. So it's not a test that we actually fully understand. There's a lot of complexity and ambiguity in the test itself. And then what about the diagnoses that don't lead to a clinical change in management? Is that a compelling reason to bring in genomics now, or is it a reason why people are sort of sitting on the fence and holding back? And then what about making predictions, where in some ways, you know, the better prepared we are forearmed is forewarned is to be forearmed, and we can use that to sort of change our general approach in pediatrics to be more preventative. But we're not very good at making predictions, and we only know certain genes like Huntington's that has a very high penetrance where we can say, you know, with certainty this will cause a problem down the line. In many other cases, we're left with perhaps risk factors or things that are very difficult to convey and translate into practice. So we have to take stock of the fact that, you know, we need more research and become better at these sort of predictive algorithms before we can implement that into practice. So I kind of wanted to tackle some of these questions today, talk about how we are now making progress with whole genome diagnostics in the NHS, especially applied to intensively ill children, how we will be thinking ahead to precision therapeutics in a project in the East of England called the Neural Net, which will help with that stratified approach to kids with neurological disorders. And then how, using a new database called the Children and Young People's Bioresource, we can begin to accumulate the data to put us in a position to better predict outcomes based on genomic linkages to real life outcomes, both health, educational, and other social outcomes. So I want to start with just a sort of an introduction to whole genome sequencing. Of course, this will sequence each of the over 6 billion base pairs of DNA in the chromosome. So it gives you the genes and it gives you the intergenic regions. We don't understand, and nor do we analyze the intergenic regions. Those will be involved in the regulation of genes. One example being thalassemia. That's a mutation that lies outside the beta hemoglobin gene, but of course will impact the amount of hemoglobin that's produced, causing a disease. That's a great example of where we want to get to with many other diseases that will lie outside the genes, but there are very few examples so far. So when we talk about even though we're getting the whole genome, we're still focusing on actionable genes, usually, the exome or, the genetic coding sequences and the mitochondrial genome. And so, but whole genome sequencing captures all of this. So we can get point mutations, structural and copy number variants. I'll show a couple in the talk. We cover the mitochondrial genome. You can detect uniparental isomy and disomy, sequence tandem repeats, hard to read regions, and these non-coding and regulatory regions. Those will be, the areas of the future, and this is where also research is needed to kind of bring into the clinical diagnostic pathway. So it really is a one-stop shop when it comes to the DNA. Of course, it leaves open the epigenome, and there's certainly other things you can think about doing, but it really is the best single test when the question is there a causative, mutation for a child's presentation? Okay, my computer has decided, it's Friday. It's probably like it's getting tired. Anyway, so we started to ask this question in Cambridge in a project led by geneticist Lucy Raymond, and a postdoc on this project was Courtney French. Can we detect genetic disease in NICU with whole genome sequencing? And so we applied this whole genome sequencing test in a relatively unselected population of ICU patients, starting in NICU and then also moving to PICU. And this is the Next Generation Children's Project that some of you will be familiar with. I just wanted to give a little bit of perspective on the project. That we were trying to cast a broad net. We had relatively few exclusions, such as feeding and growing preterms, or kids with less than 24-hour stays, such as rule out sepsis, or TTN, and kids with cancer with no diagnosis, or kids with previously diagnosed conditions. Obviously, we're not going to re-diagnose them here, but we did include conditions that are generally not thought to be genetic, such as hypoxic ischemic encephalopathy, necrotizing enterocolitis, IUGR, and then we're open to clinicians' requests. We really didn't have a very high threshold for saying no, so that may be in contrast to what you hear from the geneticist often is, they want to have a very good case for ordering a genetic test. In this case, we were really looking at what is the kind of the population incidence in NICU or PICU, and can we come up with a sense for the a priori risk of a child who might present to those units? So we wanted to be more inclusive. We also wanted to try to do this in a rapid turnaround way because we had to appeal to the intensivists, and they want to make decisions, and often they're life-threatening conditions where a diagnosis can help with escalating or de-escalating care. And so our workflow is shown here, where after having identified a potential candidate from clinical team, we would talk to the family, and we are recruiting a trio. So that's the mom, the dad, and of course the child who is consented by proxy. The sample processing was done in the NHS genetics lab. We would then send the DNA to Illumina, who did the data generation. We would do the secondary variant interpretation, feed that into a multidisciplinary team report where a decision was made, is this diagnostic or not? And then we would feed this back to genetics lab, who would do Sanger sequencing for validation, and then we could clinically return the results. And we could achieve this with a 14 to 21 day turnaround time, which we thought would help with clinical decision making. And I'm showing you here just the kind of, I guess, number of patients in our recruitment rate, which was initially 47% in the first phase of the project that incorporated 159 families. And this shows you some of the characteristics of patients coming from NICU or PICU, their age, and then you can see we started to also, at later stages of the project, started to recruit neurology and genetics patients. So this is sort of what our cohort composition looked like. They were mainly trios, 85%. In some cases, only one parent was available, or we also included some quads or siblings in some cases. And then this also shows, what is the inheritance pattern, either, inherited X-linked or, inherited homozygous or heterozygous. And then interestingly, what we see in this study and what we see in many other studies is the de novo mutation rate, where we can be reassuring to families about future pregnancies because it's a mutation that is not present in mom or dad. It's happened just in the child. And in Cambridge, at the initial phase of the study, we had 2/3 approximately were de novo. And what we have found, though, as we kind of proceed along with this study, and this will probably make intuitive sense, is that depending on the region that we're focusing on, there will be a higher or lower rate of inherited conditions versus de novo. So all comers to Cambridge, about 2/3 were de novo. But probably if we go to some other areas, such as Bradford or other areas like East London, there'd be a much higher burden of inherited disorders, reflecting consanguinity and other factors. Okay, so what's an example here? Here's a 32-week preterm male who comes in with hypotonia and contractures, requires ventilation. Multiple investigations are done, including muscle biopsy for histology, ECG, EEG, a protracted and invasive diagnostic workup that took about 6 weeks. And in parallel, at an early stage in the process, we had done whole genome sequencing, and we had a return of the genetic diagnosis of a myotubular myopathy caused by MTM1 mutation that was inherited. And we had that, well before the sort of more traditional diagnostic pathway workup had been done. And when we say it's inherited, this sort of figure shows how we determine that from our whole genome sequencing trios. We found the mutation in the child, and then here it is in the mom. So we can say that it's a sort of a dominantly inherited mutation, not present in the father. Again, we can immediately provide that for pregnancy counseling to mom. Now, this particular child had care withdrawn at parental instruction. And at that point, there were no clinical options for MTM1. But to give you a sense of how rapidly the field is changing and how new therapies are coming online, a month after the child died, there was the announcement of a new clinical trial for gene therapy for MTM1 deficiency run by Francisco Mutonia-Gosch using adeno-associated virus gene therapy. So it's another advantage of having this genetic diagnosis, even though when we order the test, we're not sure if there's going to be a clinically actionable result. We are going to have to keep pace with the progress in the field and really try to identify, these new options which clinicians or parents may want to take advantage of. So in terms of the diagnoses, this shows a number of the diagnoses that were made. As you can imagine, they'll be in all organ systems, metabolic, neurological, muscle, etc. And the chart at the bottom shows, maybe how in each case we could find that it would change our management. Because for example, in some of these, we would bring in specialists that we hadn't thought of. We could modify treatment. I'll show a couple of examples of that for seizures. We can provide pregnancy counseling, as I indicated, for de novo mutation being relatively reassuring, or for inherited dominant conditions, giving that 50% recurrence rate, but also prenatal testing if that's something the parents wanted. Also, explaining death can be very helpful, I think, in the closure that families need. And then also transition to palliative care where, conditions were deemed fatal, and that was a decision made in concert with the family. For neonates, and a real surprise was that the neonates often lack the expected sort of phenotypic features that would go along with these syndromes. So there are many cases where the geneticist would not have ordered a genetic test. We did it with these relatively permissive criteria, and then found a diagnosis because the neonates would lack the expected phenotype. So it's sort of an argument for thinking more about, applying these based on level of illness severity, and perhaps an a priori risk associated with being in NICU being sufficient reason to order a test with a relatively low threshold. And we found in the first cohort of patients that in at least 75% of cases, genome diagnosis changed care. That didn't include the diagnosis of explained death. And so I think I would sort of revise that number to say really almost in every case when we made a diagnosis, it changed care. At the very least, we mitigated the diagnostic odyssey, and we stopped testing for all sorts of other things, as often happens with kids with unexplained syndromes. So I would revise that number to 100% of the time we change care. Now, this project also, it did receive some good press coverage, because it was one of the first examples in the genomic medicine age of benefit to patients. And this video that I'll just show, which hopefully the sound will work, will give you a sense also of the impact on parents. This is about a 2-minute video.
This is life at its most vulnerable, made even more perilous when the cause of a baby's sickness is unknown. In some cases, it can take months or even years to get a diagnosis, but that is set to change because of genome sequencing. What are you trying to do to me? Millie May and her parents, Claire and Chris, each had their genome, their entire genetic code, sequenced to try to discover why she was having life-threatening seizures. The results showed Millie May has a rare form of epilepsy caused by a single gene error, not inherited from her parents. It led to immediate improvements in her care.
Since we've had the diagnosis, it's been a lot better. We had to change one of her medications due to the fact that the one that she was on was obviously aggravating the type of epilepsy that she has. We saw a big difference as soon as that change was made.
It's priceless. That one test result obviously allowed us to put all of the correct people in place and make the best for her.
Sequencing the billions of letters of DNA code that make up a genome used to be hugely expensive. Now it costs less than £1,000. Scientists in Cambridge analysed the genomes of 350 children in intensive care, comparing it to the DNA of their parents. They found 1 in 4 children had a genetic disorder and were able to give the diagnosis in 2 weeks. This study shows conclusively that whole genome sequencing is a real benefit to patients, speeding up diagnosis and helping to find the right treatment. From next year, throughout England, the NHS will offer whole genome testing to all babies and children where the cause of their illness is unknown, the first national health service in the world to do so.
Okay, so I mean, this project, I think, was helpful in raising awareness of the benefits of genomic medicine. Sue Hill, of course, really, promoting this agenda. I think the study really did help to lead to the R14 pathway coming online and some of the new genomic tests that are now available in the NHS. So now we can think ahead to how we'll sort of want to use these. Now, I was talking earlier about the a priori risk of a child coming to a particular clinic. And I'll show you a couple of pieces of data that are now helping establish that based on our own East of England catchment, kids coming to Cambridge or from around the region, because we have been now offering this to other hospitals like Norfolk Norwich University Hospital. So here's now an update, 432 trios. And now we're seeing recruitment rates of 90%. I think this is because the families are becoming more comfortable with the idea of genomic medicine. They're understanding that there have been benefits attached to this when it's an intensively ill child. And so our recruitment rates have increased to this impressive level. Now, at this particular interim stock take, we had about 1 in 5 kids that had a diagnosis coming from NICU, 1 in 3 from PICU, and the highest return rate was pediatric neurology clinic, where 42% of kids just kind of coming to clinics were diagnosed with a monogenic condition. I think a really sort of surprisingly high number, but actually consistent with what other clinics, other neurology clinics around the world are seeing. In genetics, these would be kids that had been to other genetics pathways, failed to get a diagnosis, and then had whole genome sequencing, which showed this kind of impressive return from genetics clinic. So now here's an update again. Here's 521. This is when we took a pause in the Next Generation Children's Project. So this is the extent of the data that you have. And this focuses on some of the neurological conditions. And if we focus on kids from NICU, we recruited 194 trios of those 25%. So now it looks like our final number is 1 in 4 coming to NICU with a genetic diagnosis. And then depending on what they presented with, different rates of return. With seizures, 18%. Now, I have to say, this included 20 cases of hypoxic ischemic encephalopathy, none of whom had a genetic condition. if we excluded those, we would have seen a much higher return rate for seizures. So I think this is sort of an underestimate of kids with seizures in NICU. And if we excluded HIE, those kids that continue to have seizures probably do have a neurogenetic condition. With hypotonia, surprisingly, 50% of kids coming to NICU with hypotonia, and all we excluded there was trisomy 21. So that included Prader-Willi syndrome and SMA patients. But still, if you just sort of were, I don't know, like a simplistic neonatologist, floppy baby coming to NICU, ordered genome sequencing, who doesn't have Down's facies, you might expect a very high return rate. And we saw that across ages with hypotonia. So that'll give you a sense, I think, of as we acquire more and more of this genetic information, kids coming to particular clinics, and if we're offering this test, in a pilot phase, we're really trying to understand more of the genetic contributions to presentation. We'll come up with guidelines that will help the clinician decide whether or not they think that they should activate a genome earlier or later, because you'll just have a better sense of a priori risk. Now, here are 59 genetic seizure diagnoses that we made to show that there are different presentations in different clinics or different clinic settings. NICU here would be a spectrum of disorders. PICU, some of course will have later onset, pediatric neurology clinic, and genetics. And some of these are actionable. So, for example, I think KCNQ2 would dictate a repurposing drug moving to carbamazepine as an effective pharmacogenomic approach that you can take for kids with diagnosed as KCNQ2 mutation. CDKL5 I wanted to pull out just because we've seen a couple of these. It's one of the more common diagnoses that we've seen in the East of England, and it's a very there's all kinds of crazy mutations you can see in any of these genes. You could see point mutations like this one, which is a pathogenic de novo missense. Here it is in the patient, but not in mom and dad. Here's a complex duplication inversion mutation. Of course, you could have deletions and all sorts of things. And I guess the point is that whole genome sequencing will pick up all of these and give you that pregnancy counseling information. Now, CDKL5 often missed in the first 6 months of age. It has characteristic seizures that the clinician will often miss, and a subtle EEG pattern that often the neurophysiologist will miss unless you're really familiar with rare syndrome. So it's a case where I think having genomic diagnosis can really accelerate, having genome sequencing can accelerate the time to diagnosis. So as I said, we've paused the Next Generation Children's Project with recruitment of over 500 patient trios. Overall, we've seen 25% of babies in NICU with the diagnosis, 1 in 3 in PICU, and then now 45% of children with neurological presentations coming to clinic. And as I mentioned before, we're really starting to feel that genomic diagnosis always changes care, at the very least by mitigating the diagnostic odyssey. And now we've seen the uptake in the NHS with the whole exome sequencing run from Exeter since 2020, and now whole genome sequencing, which is on the test directory in 21. And now we can talk maybe later about what are the barriers to using it, because it's still not straightforward how we can employ this. In fact, here's a message that we had from our head of the East GLH, Sarah Bowden, saying, we're authorized to do genomes, and the yellow conditions are neurological test directory indications, where you can now order a whole genome trio from gel with about a 6 to 8 weeks turnaround time. And now we're under pressure to use these, but of course, that pressure falls to the clinician, and recruiting and consenting is time consuming, and it's difficult. So, we're in the situation where now the penguin is saying, use it or lose it. If we don't start to use these tests, then maybe they're going to be repurposed. And yet, there's a whole infrastructure we need to be able to support the use of these tests, both the recruitment of families and also the delivery of diagnoses and adequate post-diagnostic support. So I'll leave that hanging. It may come back in discussion and start to talk about a little bit more how we could use this information positively for stratified therapeutics. Now, I think genomics will have a role in the assessment of a number of neurological conditions. Cerebral palsy is one that we want to now start to develop pilot data for in the East of England. We also have a relatively poor understanding of the extremely low birth weight preterm infants who we know are vulnerable to injury, IVH, etc. there may or may not be a genetic contribution to the cause of prematurity, an open question that these tools will also help us with moving forward. But here's a real actionable one, and this is where you'll see a lot of focus internationally is on epilepsy presenting in NICU. And there are about 2,000 such children in the UK each year who will have epilepsy. So it is the most common neurological emergency in NICU. It is challenging to diagnose. It has a varied prognosis and treatment depending on the underlying cause, but I can sort of, I guess, confess that as a neonatologist, we would just treat with one drug. We always reach for phenobarbital or Keppra, depending on the particular preference of a site. And so we're not treating things with respect to the complexity of the underlying condition. While I was head of neonatology at UCSF, we did start a stratified kind of diagnostic approach to kids with seizures, which we call the neurointensive care nursery. And that model's been adopted by TOP in Austin, the East of England, as a regional program to serve the 1.5 million children or 80,000 deliveries per year, of which 240 approximately are predicted to have neonatal seizures. This shows TOP in on the right with a MRI compatible incubator and the donors in the background, which we now have at the Rosie Hospital. So that's part of the kit that you'd want to use to evaluate seizures. Another is, you want to be able to look at a baby like the one upper left, who looks like he's sleeping, who has a SAT of 98 to 100%, and just be sure, does he have seizures or not? Now you can see he's on a cooling blanket, so that's a tip-off, and the EEG is in the lower right-hand side. So that is a baby with status epilepticus, and it highlights why in NICU we need to have EEG to diagnose a seizure, because there are many subclinical seizures, and there are many things that mimic seizures that don't have an electrographic correlate. So a big push needs to be made to be able to bring EEG into NICU with continuous video EEG monitoring during hypothermia and in our fuller assessment. So that would be one of the things that is needed. And again, we need to work to develop that resource. The MRI compatible incubator gives us a much better resolution scan. We want to be able to screen for metabolics, and then we want to use genomics. And that can help us determine, is it a brain malformation, like a segmental defect? Is it HIE or stroke? Or is this a rare genetic, such as KCNQ2, for which we would use that repurposing drug? And Roberta Cilio, who would be a great person to invite to a future talk, Ben, is really one of the world's experts in neonatal rare epilepsy syndromes, diagnosis and treatment, and was one of the first to propose repurposing carbamazepine for use of KCNQ2 encephalopathy. If anybody has seen the clinical impact that this can have, it's dramatic. A kid who might be comatose on multiple anti-epileptics, who then, goes on monotherapy with carbamazepine, wakes up, breastfeeds, goes home, and it really is a great example of how we can use this data to improve therapy. And Amy McTague in the UK is really one of, I think, the leaders, focusing on this, and how can we use the genetic diagnoses to use to program certain anticonvulsants and ones to avoid. And you heard about from Millie May's parents, so Millie May has an SCN1A mutation, and she had been on carbamazepine, which was stopped, and that actually improved her seizures. So this will be a good example of how we will use genetic data for pharmacogenomic repurposing and more stratified treatments. And we'd like to think that in other conditions, there'll be similar types of information available. So matching genes to medicine will be, I think, a direction forward for research. Then another area is sort of just discovery, so it's impressive that we can diagnose 42% of the kids in neurology clinic. What about the others? There's a lot more to learn, and we want to be able to, bring in assets, and Cambridge and East of England have a lot of those to be able to look at both how we can use, become better at DNA diagnosis, but also how we can interact with basic science colleagues in places like Laboratory for Molecular Biology to determine the mechanisms of variants of unknown significance to help us, convert them to a actual diagnosis that we can deliver. I won't have time to talk about that, but there are actually a number of examples we can cite from local researchers that show that we're interacting well with the basic science colleagues to be able to do functional genomics, take information like VUSs from the clinic with a detailed clinical phenotype, put them into models, diseases in a dish models, and then come up with mechanistic insights from those mutations that we can then feed back to clinicians. Sorry about the noise. I live near Addenbrooke's, and it sounds like there might be a helicopter or plane just landing. So then moving on to the third part, which is this predictive testing, and this will be pretty speculative, and it will kind of highlight, I think, where we're also going to try to further exploit opportunities in genomic medicine, looking forward to a new type of database that will link genomic information to long-term electronic records as a proxy for outcome. So let's imagine 20 years ahead, we'll even be doing whole genome neonatal screens. Some of you may have been in the consultation because this is being promoted by GEL currently. Should there be pilot projects for neonatal screens using whole genome sequencing? That will raise a lot of concerns, and so I'm not going to dwell on that, and I'm just going to sort of bypass that discussion and say, well, let's say, but in 20 years we'll figure that out. So 20 years we'll be doing this, and we'll be detecting things much earlier, and we'll want to use that information to make better clinical decisions. But there's a big knowledge gap, and so we're going to need to do more than just sort of do whole genome sequencing. We're going to need to see how a baby born with a particular genetic finding links to outcomes. And because the UK does have national records for health, education, and socioeconomic proxies, we have a unique opportunity to be able to link genetic data to those outcomes and kind of consider a living database where we're learning as we become better at putting these into practice. And that's the inspiration behind a new Bioresource branch called Children and Young People Bioresource. And the concept here is that we can recruit diverse populations of children and young people, depending on the study, different biomarkers. Some would be DNA, or there may be imaging or other biomarkers. There may be opportunity for patient input, or that could be from a Fitbit or from just self-reported information. We can link then to various records such as health records or the educational records. Interestingly, Department of Education does have a interest, I think, in using some of these linkage techniques to be able to come better at predicting children at risk for learning disability, or maybe approaches to therapy that could be better for them. The updating is going to be required because even when it comes to a genetic diagnosis, next week things might change, and one of these variants of unknown significance might convert to a diagnostic just because other kids with that genetic finding have been reported. And so we need a way of just being able to take the data and always refresh it. By coupling to long-term outcomes, we try to achieve the following clinical goals. Can we shift from reactive medicine to more disease prevention? Can we start to look at the causes of disease? When it's genetic, we'll have a good sense for that. Or if we think it's more environmental or a balance, maybe a child in a vulnerable situation with a genetic susceptibility, and we can come up with a better understanding of causes of disease and how we might be able to build resilience for that child. And then maybe the low-hanging fruit, or where we'll be focused for the next 5 to 10 years, better diagnosis, and then how we can use that to target therapies. Where this database is different than something like Bourne and Bradford or ALSPAC or retrospective databases is that we will be acquiring data in real time, and if we can provide the right medical investment and support for studies such as the Next Generation Children's Project, which had geneticists and intensivists involved, we can take advantage of the fact that this is prospective, that it works sort of in real time, and we can make changes in management. We can suggest medical treatments. We can recruit into clinical trials such as gene therapy trials. And so we're not just learning from, but we're also benefiting. And I think if we can keep in mind these, the orange principles, this will be a database that's very different than most of the other ones that are around looking at genetics, because it really is going to try to provide impact and evidence of impact as we go along. And I think that will be key for helping the clinician and families understand that these tools are beneficial, that they are helpful and should be incorporated, versus skepticism and focusing on the concerns around these techniques. Now, the neural net I mentioned before is a a new project that will initiate sort of in the fourth quarter of 21. And this is going to also try to take advantage of some of the unique in Cambridge and East of England, where we're going to look at improving diagnosis in the NHS, but also, again, taking advantage of these science assets with making, patient models of disease, research into novel therapies, and then tracking those and associating them with outcomes through the Bioresource approach. So NHS and improving diagnoses. Now we're taking the Next Generation Children's Project bioinformatics pipeline and embedding that in the East GLH, which will serve East of England and East Midlands. So that's a good example of how the research will directly move into practice. Of course, we're going to need to get ISO accredited for that, and that will be the work of the next few months. And then when we have those VUSs, we'll have university research looking at improving our diagnostic capability through bioinformatics, but also those sorts of mechanistic studies that we can do using in vitro models or animal models. Now that ideally will lead to new ideas about precision medicine, and we want to bring industry along. There is a rare disease industry that has interest in these conditions, so there may be opportunities to work with them, and then measuring the outcomes, which would involve the children and young people bioresource. So just to give you a sense of this approach that we're taking to try to deliver on the challenges of how we take a prospective database and we can turn it into benefit and be sure it's beneficial or have a sense, because we're going to follow kids using some of the national records that are available. That's the concept. And this will start in the East of England. And so the ambition would be to apply these tools and this genetic diagnostic capability, not just in Cambridge, but from Peterborough, from Norwich, from Colchester, working with ARU and University of East Anglia, and really, make this a regional resource. And again, we can talk about the opportunities and barriers for uptake. But I know, Ben has an interest, and I think a lot of clinicians are keen to start to employ these tools, and yet, there are some practical barriers that make that difficult. So I'm starting to wrap up here. So I've told you about whole genome diagnostics that are useful in, conservatively 1 in 5 to 1 in 3 children in ITU. The thinking, towards development of stratified therapies that is encompassed by the Neural Net project, and our plans for, developing this kind of living database to also learn from and enhance care. Now, the Children's Hospital, people will be aware of, is on the horizon, and I wanted to just highlight a couple of the ways that Children's Hospital will help to deliver as a regional, asset some of these goals. And you could kind of think of this as a model hospital in the sense that it will be driven by 2 ideas that will really almost be USPs. One of the great things about this project is the ability to look at how we can treat the whole child, and rather than treating physical and mental health in silos, we can look at true integration. And that's because this is a collaboration between 2 trusts, CUH and then Cambridge and Peterborough Foundation Trust, where, because they need rehousing of the Tier 4 facility in Fulbourn, all coming together on the Edinburgh site, we are looking at blended clinical wards with physical and mental health care with minimal segregation, in some cases, absolutely joint care by both teams, with psychiatry and acute pediatrics on the same floor to really enhance an approach to children with complex comorbidities. That's based on data. This is a paper from UCLA that shows children admitted to UCLA hospital with complex comorbidities, either a primary medical diagnosis like cancer or inflammatory bowel disease, but then who have developed a psychiatric comorbidity like anxiety or depression, and that's the black line, or a primary psychiatric diagnosis like eating disorder, who then have medical complications, and that is this line here. And that's in contrast to psych only that are treated much more in community versus inpatient. So what do we do for those patients? This is also what we're seeing in Cambridge. You're probably seeing it in Colchester, and you're seeing it in any place a child is admitted. We're seeing a lot of kids that have mental health problems or self-harm, and we need a model to deal with that. I think the right model is to have psychiatry and acute pediatrics working together in one place, and that is what this hospital aims to model, is the integration of physical and mental health care. And then what I sort of affectionately term the new pediatrics, which is how we look ahead and we start to think about a baby for the adult they will become. We'll have a lot more information about the health risks. We know that many chronic diseases, not just rare disease, but common disorders like diabetes and heart disease, or even dementia, mental health, will have their origins in children, young people 0 to 25 years of age. So if that's what pediatrics is going to be, if that's what we'll be treating as pediatricians, according to the NHS long-term plan it is, can we start to really look at the origins of physical and mental health and use that to program preventive interventions and, come up with an antidote to the traditional, wait for something to break and then you try to fix it approach. For serious genetic disease, we have an opportunity there. That might be one of the places that we start, but of course, many other opportunities to think about how we could structure our research to look at origins of obesity, diabetes, heart disease, and so on. And that's how our thinking will also be channeled in our research priorities. Demonstrate that we can use early diagnosis to prevent and then decrease disease burden. And this would be then improving physical and mental health over the life course and starting early rather than in middle age. We start in children to maximize the lifelong impact. So that's another key idea in the research that would occur within Cambridge Children's Hospital, which will have a University of Cambridge Research Institute. So it's a hospital that sort of has a promise. We have the hospital and then Cambridge Children's Research Institute, the promise to advance child health. We aim to be the first hospital of its kind, which is purpose-built for integration of physical and mental health care, and that will fuel research. And it will be underpinned by cross-cutting technologies, including imaging and genetics. We will aim to function as an effective regional hub to support community practices. Providing retrieval, you'll be aware that the PANDER service has been stood up now. So now we have regional coordinated neonatal and pediatric retrieval. And so this, I think, is going to mean better care, more coordinated and closer to home for families, saving trips to London or out of region. And we're evolving a digital strategy to promote, telehealth and better coordination between DGH's Cambridge and community practices to help that. On the research side, our data strategy will involve that new children, young people bioresource. And then our research, which we're looking ahead to come up with very innovative approaches to look at origins of disease as a way of coming up with rational therapies and thinking ahead to prevention. The other, in purple, unique feature and really, I think, significant challenge we have is that the NHS has challenged us to raise $100 million for the hospital through fundraising. We think that's possible because the University of Cambridge is involved, and this will be cascaded throughout the International Alumni Network. But and we're starting to see some progress. The campaign launched in March this year, and you never know, we may make it there, but certainly any help would be appreciated in that very unique challenge that this hospital has. And of course, we're also looking ahead to the next generation, and these would be just some of our trainees coming up through the pipeline who we think will be academic leaders and help to expand and diversify our research activity going forward. I'll highlight one, Thaloka Radnicki, who will be probably based at Ipswich. And so, again, we're thinking these are trainees coming from Cambridge, but instead of them all getting kind of sucked into the vortex of London, can they go to Norwich? Can they stay in the region? And maybe some of their research will be supported from Cambridge if they're academically inclined, but they're working, around the region, not just focused in Cambridge, and we don't lose them outside the region because we have some really talented young people. So 2025-26 is when we are expecting this hospital to open. I'll finish with a few acknowledgements for the Next Generation Children's Project. Lucy Raymond, I mentioned, who was leading, David Bentley from Illumina, Toppin from NICU, and then moving forward, these are some of the people that we're talking to about the neural net project. I went a little bit over time, Ben. I'm apologize.